Hello, everyone, and thank you for joining. I'm Brad Evans. Today, we'll dig into the scientific drivers that determine whether biomarkers succeed or fail, not at the assay level, but at the biological and specimen level. Across precision medicine, biomarkers often fail due to hidden pre-analytical and contextual factors that distort biological signals. This talk will walk through those mechanisms and highlight strategies to design biomarker studies that more faithfully reflect the underlying biology. Let's start with the scope of what we'll cover today. This session will focus on 5 major scientific themes. First, why biomarkers often underperform even when the biology is sound, typically due to sample-driven artifacts rather than assay design. Second, how biospecimen integrity, tube chemistry, processing times, and contextual metadata directly affect biomarker reproducibility. Third, what fit for purpose truly means as we move from discovery to clinical assay development. We'll also examine the pre-analytical constraints imposed by multi-omic and spatial platforms, particular cell-free DNA, cell-free RNA, extracellular vesicles, proteomics, and high-plex tissue arrays. Finally, we'll close with practical design strategies using matched and longitudinal samples to support more rigorous biomarker interpretation. With that foundation, let's anchor ourselves on why biomarkers matter in the first place. Biomarkers underpin every stage of precision medicine. The graphic on the right shows a continuum from target identification through post-approval monitoring, where biomarkers function as mechanistic readouts, patient selection tools, and indicators of pathway modulation. Scientifically, these fall into 4 categories: prognostic, predictive, pharmacodynamic, and diagnostic biomarkers. Each category reflects a distinct biological phenomenon. Prognostic markers capture underlying disease aggressiveness independent of therapy. Predictive markers identify biological actionable pathways. Think the PDL1 story or oncogenic drivers. Pharmacodynamic markers quantify on-treatment biological shifts such as cytokine kinetics or ctDNA decay. Diagnostic markers anchor disease classification, like HER2 or ALK. The key scientific point for all of this is that these biomarkers are only as reliable as a biological signal preserved within the specimen. If upstream conditions distort that signal, downstream interpretation becomes fundamentally compromised. Those scientific dependencies help explain why biomarker programs experience such high attrition. This slide outlines the primary scientific barriers that derail biomarker programs. The biggest is pre-analytical variability. Tube additives, time to spin, shear forces during centrifugation, and thermal excursions all alter molecular composition. For example, delayed processing causes leukocyte activation, releasing genomic DNA that contaminates CF and ctDNA measurements. Extracellular vesicles rupture with high G spins. Proteins denature during freeze-thaw cycles. Next is missing clinical metadata. Timing relative to dosing, steroid use, performance status, imaging intervals, and response to treatment are all important variables that need to be addressed. Without these covariates, even robust biological signals become uninterpretable. Third is assay reproducibility. Discovery workflows often depend on pristine, low heterogeneity specimens that don't reflect real-world variability. Finally, lack of matched or longitudinal samples and poor translation to heterogeneous populations limit biological validity. To address these challenges, we need to improve the biological fidelity of the specimens themselves. This slide breaks down the mechanisms through which pre-analytical factors disrupt biological signals. Tube chemistry affects cellular stability, nucleic acid fragmentation, and protein oxidation. EDTA, Strec, and packaging each preserve different biochemical states. Processing delays cause hemolysis and introduce contaminant genomic DNA and degrade extracellular RNA. Centrifugation parameters determine which extracellular vesicles remain intact, whether certain RNA species pellet or remain in supernatant, and whether proteins remain in complexes or dissociate. Freeze-thaw cycles further damage proteins, lipids, and vesicles. The infographic at the bottom quantifies how QC failures cascade into assay failures and lost time, a scientific demonstration that upstream integrity directly controls downstream precision. Building on that, we need to design biomarker workflows based on biological, not operational requirements. Biomarker development is inherently nonlinear. The clinical question must define the sampling strategy, specimen type, and analytical platform. From a scientific standpoint, you must ask, does the analyte exist in the chosen fluid at measurable levels? What sampling window captures meaningful biology? Will biological kinetics differ pre versus on treatment? Is the assay platform compatible with the biochemical state of the specimen? And ultimately, does this workflow align with the intended scientific decision? The diagram on the right emphasizes that sample strategy, detection method, validation path, and intended use are biologically connected, not independent choices. Next, let's examine what constitutes an insight-ready biospecimen. This slide outlines 4 scientific pillars required for high-fidelity biospecimens. First, ethically sourced regulatory grade acquisition is essential not just for compliance, but for reducing biological variability. Consistent fixation, draw order, and processing conditions improve reproducibility. Second, clinically rich annotation provides the context needed to interpret biological signals. Disease stage, treatment history, timing relative to therapy, and imaging data serve as essential covariates. Third, matched and longitudinal sample sets allow cross-compartment comparison, tumor to blood, immune microenvironment to systemic immune activation, and allow us to quantify kinetics rather than rely on static snapshots. Fourth, compatibility with multi-omic platforms requires preserving nucleic acids, proteins, vesicles, and tissue architecture in a way that supports integrated analysis across platforms such as RNA-seq, proteomics, spatial biology, cfDNA, and extracellular vesicles assays. Now let's look at how these principles play out in immuno-oncology specifically. This slide shows how tumor and peripheral blood compartments provide complementary biological information. The tissue images, H&E, CD8 staining, and PD-L1 expression demonstrate local immune infiltration at checkpoint biology. The genomic profile on the right lists multiple co-occurring mutations and a RET fusion, illustrating the complex genomic background that shapes local immune contexture. When pairing with PBMC profiling functional assays or systemic cytokine measurements, these cross-compartment datasets illuminate how local tumor biology aligns with or diverges from systemic immune activity. Scientifically, this integrated approach reveals the resistant mechanisms, identifies actionable pathways, and supports more predictive biomarker development. To understand biomarker dynamics, however, we need longitudinal sampling. This slide focuses on biomarker kinetics. Longitudinal sampling is uniquely powerful because it captures biological trajectories rather than static snapshots. MRD tracking relies on percent change in ctDNA allele frequency, not absolute levels. Similarly, resistant evolution requires repeated sampling to identify emergent variants. Pharmacodynamic biomarkers reflect pathway modulation over time, and cut point determination isn't possible without repeated measures. The line graph on the right illustrates how patient molecular burden fluctuates across treatment cycles, demonstrating how kinetics provide richer insights rather than single time points. The specimen strategy on the right emphasizes that different compartments, tissue, plasma, PBMCs, CSF, each contribute complementary biological information. And I want to highlight the figure on the bottom left, indicating that the time that you would measure the biomarker would change what biomarker is important, and so it's very important to have that longitudinal sampling. As biomarkers move towards clinical use, upstream rigor becomes even more important. Although the language here refers to regulatory expectations, the deeper point is scientific. Analytical validity is inseparable from biospecimen validity. Histopathology ensures diagnostic accuracy and biological representativeness. DNA and RNA integrity values quantify the nucleic acid integrity. Protein concentration and degradation markers confirm biochemical fidelity. Controlled storage and shipping prevent temperature-induced biological drift. Whether or not a biomarker is intended for regulatory submission, the scientific principles behind regulatory-grade collection directly influence reproducibility, sensitivity, and effect size detection. Next, let's turn to how multi-omic and spatial biology approaches depend on upstream rigor. This slide describes the multi-omic integration pipeline. FFPE and fresh frozen tissue support RNA-seq, proteomics, spatial transcriptomics, and multiplex IHC and immunofluorescence. Each modality imposes distinct pre-analytical requirements, whether fixation time, section thickness, and ischemia delay all influence RNA and protein integrity. Digital pathology enables quantitative, standardized inputs for AI models. Machine learning amplifies both high-quality signal and pre-analytical noise, making upstream consistency essential. The workflow diagram shows how integrated data sets, gene expression, protein abundance, spatial immune features, all support biomarker discovery, predictive modeling, and translation to regulatory-grade assays. With these platforms in mind, let's examine the role of real-world evidence. This slide illustrates how real-world biospecimens illustrate biological diversity. Age, comorbidities, and prior treatments all better reflect clinical populations. Scientifically, this diversity exposes batch effects earlier, sharpens cutoff refinement, and tests robustness across heterogeneous contexts. The diagram of reoptimization loops illustrates how assays often need recalibration once exposed to realistic biological variation. Real-world samples therefore accelerate translational readiness by validating biomarker performances across a full spectrum of patient biology. All of these elements come together in the broader evidence pipeline. This slide shows the continuum from discovery to validation, clinical trials, and therapy approval. The scientific message is that each stage depends on the upstream biological integrity. Fit-for-purpose biospecimens reduce noise in discovery, improve reproducibility in validation, and enhance predictive accuracy in trials. Real-world evidence then identifies resistance, establishes durability, and supports refinement. This pipeline reflects a biological truth. Biomarkers succeed when specimen quality, analytical rigor, and clinical context are aligned from the start. I'll briefly acknowledge the last few slides, which summarize broader considerations. This slide summarizes the scientific infrastructure needed to support biomarker development across discovery, translational research, and diagnostic applications. The scientific message here is that high-value biomarker studies rely on specimens that capture real patient biology. That includes sufficient clinical depth, including treatment history, outcome data, and imaging, to correctly interpret molecular signals. It also requires biospecimens collected under consistent, well-documented conditions so that the biology measured reflects true disease biology rather than pre-analytical artifacts. And finally, scalability matters. The same biological integrity should hold whether the specimens are used in early discovery assays or highly stringency clinical validation workflows. Together, these elements create a scientifically reliable foundation for biomarker interpretation across multiple research settings. Next, let's briefly review how different specimen types support different scientific applications. Now I'd like to talk about how BioIVT can support your biomarker work. This slide shows our location and diversity of biospecimens offered to support your biomarker studies. It is also showing the types of assays supported by the different types of biospecimen. Tissue, both FFPE and fresh frozen, can support spatial biology, RNA-seq proteomics, and multiplex IHC because it preserves architecture and molecular gradients. Plasma and serum enable cfDNA analysis, cytokine profiling, and proteomics, where processing speed and tube chemistry directly influence analyte stability. CSF provides access to CNS-restricted biomarkers like some of the tau species, synucleins, which are often not measurable peripherally. PBMCs allow detailed immune profiling, receptor sequencing, and functional assays, while fluids like urine or BAL are useful for exosomal and metabolic studies. Matched tumor blood sets and longitudinal sampling add an additional layer of biological rigor, enabling researchers to connect local tumor biology with systemic markers and to track kinetics over time. Now I'm going to close briefly with a summary. To wrap up, biomarker success depends on biological signal quality, controlled pre-analytical conditions, and the alignment between specimen type and the scientific question. When specimens are well annotated, preserved appropriately, and collected in a way that supports multi-omic analysis, the resulting biomarker data are far more reproducible and clinically meaningful. Thank you for your attention, and I'm happy to discuss any of these scientific considerations further. This concludes the talk. Thank you.